Hi, this is me again, and for this video, I'm gonna talk about senses starting with taste and smell. And these senses are pretty unique because compared with the other senses that we have in our body, um, these are gonna be chemical. So they're gonna have chemical receptors. Most of the other ones have mechanical receptors, and that's why they are pretty unique. And I'm going to explain how taste works, how we convert that chemical signal, we take it to our receptors, and finally we take it to our brain to be converted to something that we can understand. Okay, so we're going to have five major tastes um, in our tongues, being salt, sour, sweet, bitter, and pretty recent discover umami. Umami. I don't know how to pronounce it. Within our tongue, we have these structures that we're gonna call taste buds. Okay, and now I'll explain how they work. Starting with salt. So the cool scientific name for salt is sodium chloride. When we eat something salty, it's gonna be dissolved by our saliva, and we're gonna separate and create two different ions, sodium and chlorine. We're gonna have an E sodium channel that is gonna be open 24/7. Okay, so this is not going to be dependent on voltage or ligand or something. So this is gonna allow salt to come in. As we know, salt likes to be inside of cells. Okay, their driving force is to be inside of cells. So they're gonna come in. Okay, and pretty similar compared with the other examples that we have learned in class. This is gonna make the cell to depolarize. Okay, so that means we're gonna make it more positive, and those positive charges, as we know, the inside of the cell is negative, the outside is positive, is going to depolarize the cell and allow calcium voltage gated channel to open. Okay, we described previously how the channels work. And again, the driving force of calcium is gonna take some calcium from the ECF to the ICF. Okay, so now we have calcium within the cell. The cell is going to depolarize even more. Something important to say, when we talk about tastes, we are never going to reach an action potential. They're gonna work with something different named gradient potentials. We already know what is that. It's something more to do with the concentrational gradient driving forces, what likes to be inside and outside the cell. So in this case, the entry of calcium within the cell is gonna facilitate the exocytosis of neurotransmitters. In this case, well, we know the mechanism. They're gonna attach to the SNORD proteins, making the proteins to have a conformation and then releasing the neurotransmitters. For this one, we're gonna have serotonin or ATP. Okay, so and that's pretty much it. What's gonna happen then, we know, is gonna be triggered by a receptor, and that's gonna go to a, a brain cell and take it to the brain to be translated. So that's a little bit of process, the summary of how it works. And now I'll describe how sweet, umami, and bitter tastes work. And they're gonna have pretty much the same process, okay? So compared with salt, we're gonna have a G protein. As we know, G protein has a receptor that is gonna be triggered by a ligand. So in this case, these ligands are gonna be carbs, quinones, or glutamates. So those chemicals that are gonna trigger the G protein. After, we're gonna have that alpha subunit leaving the G protein, attaching to phospholipids C, also named PLC, and we're gonna create an IP 3. It's gonna act pretty similar compared to the mechanism in our muscle. The IP3 is gonna go to the opening of the cytoplasmic reticulum, making calcium to go out of the cytoplasmic reticulum. Since we have a greater concentration of calcium in the inside of the SR, it will want to go to the outside, to the cytosol, and the process is going to be the same. Calcium is going to attach to the SNOR proteins. The SNOR proteins is going to have a conformation, and then the neurotransmitters are gonna be released to the synaptic cliff by exocytosis. Okay. Now, sour. Sour, it has to do with acidic things. Okay. Yeah, I was looking that I was not saying something wrong. So sour, again, we're gonna like dissolve those hydrogen ions by our saliva when we eat, and within the cell we're gonna have this hydrogen channel allowing hydrogen to come in. Hydrogen has a positive charge, so as we know, what's gonna happen is going to depolarize the cell. Compared with the others, we're gonna have a potassium leak channel, and this leak channel is gonna close. So we're gonna block the entry, the exit of potassium to the ECF. So again, we're making the cell more positive. We're depolarizing it even more and more, since we don't have more positive charges leaving the cell. So it's gonna happen pretty much the same. Now, these positive charges are gonna open this calcium voltage gated channel, and we're gonna have calcium getting into the cell, attaching to SNOR proteins, and we're gonna have exocytosis of receptors. So that's pretty much how it's gonna work. Now, I'm gonna go through smell. And this is gonna be that was wrong. Okay, so we have our nose. Again, our mucus is gonna help us to facilitate this process, and we're gonna have these nerves in our nose that are gonna go from our nose cross this bone structure named the cribriform. Cribriform is funny because it looks like a crib, if you haven't seen it. And we're gonna take this olfactory neuron, that signal to the brain. Okay, so how's that gonna work? Okay, we're gonna have our good friend G protein, as we know, that is triggered by a ligand, other words, a chemical. So that chemical is gonna trigger the receptor. Then the alpha subunit is gonna leave the G protein, is gonna trigger adenylyl cyclase. Adenylyl cyclase, I'm gonna draw it here. We're gonna add some ATP to adenylyl cyclase, and we're going to release cyclic AMP. So that means that we are gonna break apart two phosphates, and this cyclic AMP is gonna trigger an ion voltage gated channel, facilitating the entry of positive ions, as they are sodium and calcium. Okay, because we know that they like to be inside the cell. Okay, so what's gonna happen now? We're gonna have this voltage gated chlorine channel. We're going to depolarize the cell. making this voltage gated chlorine channel 2 open. And driving force of chlorine, it doesn't like to be in the ICF, it's gonna go to the ECF. So we're going to depolarize even more the cell, and compared with taste, we're gonna actually reach an action potential in our brain. And this is like, as you can see, it has a really short pathway compared with other senses. And also is gonna trigger a part of our brain. Well, it can trigger multiple parts of our brains, the signal, but one especially is gonna be the one that is related with our memory. So that's why usually when we smell something, we can have like that memory is triggered. Like it's like a response of our body. Like for me, something that reminds me home is the smell of homemade beans. So every time that I smell homemade beans, it's like going back to my home. It's interesting. So that smell, that taste, because they have long processes. I'm just gonna invest my time for this video to explain these two senses. And for the next video, I'm going to explain how vision works. That is going to be a little bit more complicated. And finally, how we hear. And that's it. Nefi Varela, I hope you like it.